Hi everybody. In this talk I'm going to be speaking about individual participant data meta-analysis for healthcare research. Throughout the talk my focus is on the basics, so the key concepts of what IPD means, what IPD meta-analysis projects are about, why we may embark upon them compared to doing a traditional meta-analysis of published data, the advantages and challenges of having IPD, give some notable examples of success stories in IPD meta-analysis projects for treatment effects, for test accuracy, for prognostic factors and risk prediction models, outline the key steps involved in IPD meta-analysis projects, and showcase some new resources and further reading, including our new textbook, Individual Participant Data Meta-Analysis, a handbook for healthcare research, published by Wiley in 2021, and our new website, ipdma.co.uk, which showcases resources and key learning materials for researchers new to the field. So part 1, the need for meta-analysis. So in healthcare and medicine, when we're making decisions about how to treat people and how regularly to follow them up and monitor them, for example, we should be wanting those decisions to be evidence-based. And that means we need to be putting together the available evidence, or at least the higher quality evidence, to come up with findings that guide those decisions. Single studies may be subject to chance, they may have low power to answer the research question of interest, and they may even disagree in their recommendations. And therefore, the idea of a meta-analysis is to put the results together, to reduce the play of chance, to increase our power, and to come up with some clearer recommendations for those that have to make decisions, including doctors and patients. For the last 30 years, we've seen a huge drive towards systematic reviews for this purpose. So systematic reviews are an approach that uses transparent methods to identify, appraise and combine a body of research evidence. And the aim is to produce summary results that guide best practices for key stakeholders. And Cochrane are a leading example of undertaking systematic reviews, traditionally based on randomized trials to evaluate treatment effects, but nowadays in the Cochrane library we also see systematic reviews of diagnostic test accuracy studies, prognostic factors, and the performance of risk prediction models. Meta-analysis is from the Greek, which means after, above, transcending. So this is the statistical analysis that usually accompanies a systematic review to combine the quantitative results from the studies identified. A broader term is evidence synthesis. Meta-analysis allows us to quantify effect sizes such as treatment effects, the accuracy of a test, the performance of a prediction model, and also the uncertainty of these estimates, to produce summary results, again to inform the decision-making process of interest. So if we're combining results across studies, we're hoping to reduce the problem of the play of chance. We may be interested in what's causing genuine differences across studies, for example in the treatment effect. This is known as between-study heterogeneity, and we want to observe that across different settings. We might be interested in trying to personalise treatment decisions or care. For example, what factors modify treatment effect, known as effect modifiers or interactions. But we also may be interested in summarising the quality of existing evidence, so whether there's publication bias, whether there are concerns about the reporting standards, the analysis methods, whether participant groups are represented or underrepresented, if they're missing outcomes, what are the further research needs. Meta-analysis is very, very important to summarising the existing evidence and also establishing the current state of play in the particular field of interest. Part 2. Traditional systematic review and meta-analysis framework using aggregate data. So traditional systematic reviews and meta-analyses use aggregate data. So by aggregate data we mean information that's been averaged across participants in a particular study, and their results and summaries that are presented in publications or obtained from study authors directly. For example, reviews of randomized trials evaluating a treatment effect will extract information about participant characteristics such as mean age, proportion female, the study design, randomized trial, whether it was clustered, multi-center, for example, the analysis methods, for example, whether logistic regression was used, Cox regression, outcomes of interest, and key results, such as the estimated treatment effect for that trial. So this is typically going to be a measure such as an odds ratio, a relative risk, or a hazard ratio, and the uncertainty around that estimate. So a standard error, the variance, and more broadly a confidence interval, depicting the uncertainty of your estimate of interest. So aggregate data is a summary of the information about the participants in those trials and their follow-up and outcomes. Here's an example of aggregate data collected from 10 randomized trials evaluating the effects of antihypertensive treatments. So patients recruited with high blood pressure, hypertension, and they're looking to see whether this antihypertensive treatment is reducing blood pressure over time. And so we have a summary of information for each trial. So we have in our dataset a row for each trial. I'm just showing rows 1, 2 and 10, omitting rows 3 to 9 for brevity so you can see more clearly. We have the number of participants in the control and treatment group for each trial, the mean age, here it's 42, in this trial 69, so slightly older. The baseline characteristics, so prognostic factors at baseline, for example the mean blood pressure in each group before treatment. The follow-up information, the mean blood pressure at 1 year in each group, and the treatment effect. So here the treatment effect has been calculated after adjusting for baseline, and we have negative values, which in this particular situation indicates that the antihypertensive treatment is doing better than control at reducing blood pressure by 1 year. So what are the advantages of aggregate data? Well, in theory it should be simple. You just go to a publication and you extract the results. So there's no reliance on obtaining new data from participants, and in theory, if studies are clear and completely reported, we should be able to extract those results relatively quickly and cheaply, at least in comparison to a new trial, or even later we'll see an individual participant data meta-analysis. Meta-analysis methods, which summarize the aggregate data, are also quite well established. So you may be familiar with methods such as the inverse variance approach, where we do a weighted average of for example, the treatment effect estimates across studies under some assumptions such as a common effect or a random effects model, which I'll come on to later. Software for doing meta-analysis is well available. For example, RevMan, which is freely available in Cochrane, Metaphor in R, AD, Metan or Metan in Stata. These all lead to nice graphical displays such as forest plots which summarize our results. For example, let's go back to our meta-analysis of 10 hypertension trials. In these trials, remember we extracted the treatment effect estimates, and these indeed are all negative as mentioned for each of the trials. We then pull these together in a meta-analysis model where we do a weighted average across these, so weighted by the precision of the studies, so the bigger studies, the ones with the smallest variances and the narrowest confidence intervals, have the most weight. And the average result at the bottom here is our meta-analysis results. So the centre of the diamond, minus 9.84, suggests that antihypertensive treatment on average compared to control reduces blood pressure by 9.84 millimeters of mercury more. The confidence interval is again all negative, very far away from zero. Strong evidence from this meta-analysis that on average antihypertensive treatment is effective, and this is based on the aggregate data available from those trials and is a nice summary of the available evidence. What are some potential disadvantages of using aggregate data? Well, you're reliant on the reporting of published articles, so you're often faced with poor reporting. So p-values may be given rather than estimates, confidence intervals may not be reported. You're not in control of the analysis method, so it could be that you wanted to pool hazard ratios and account for censoring and time to event, but the original authors used a logistic regression model, so you only have the odds ratios rather than the hazard ratios. You may want to produce conditional treatment effects or adjust for prognostic factors, but the original analysis may not have done that. The analysis may even have been completely wrong before, so they may not have accounted for clustering, they may not have examined non-proportional hazards, they may have looked at continuous variables but dichotomized them rather than looking at potentially non-linear relationships. A meta-analysis of aggregate data is also very vulnerable to publication bias. So that's where some studies with significant results are more likely to be available to you and give you their aggregate data than those of non-significant studies. Similar issue is outcome reporting bias, where studies will report the aggregate data for outcomes that were in some sense significant, but not for outcomes that were not significant. It's also very hard to go beyond the original analysis, so you're basically reliant on what people did before, and you couldn't, for example, suddenly want to look at continuous covariates in a nonlinear way or examine non-proportional hazards. You can't do that with the aggregate data. A major issue is that the aggregate data is summarizing the participant level information, so you're collapsing participant level data down to an average for that study. So you observe mean ages, proportion male, the overall treatment effect. So this means you've lost power to explain participant or individual level variation. So you couldn't, for example, include or examine prognostic factors. You couldn't identify subgroup results and examine potential effect modifiers or interactions. You can't look at whether some patients are doing better than others because you haven't got information at the patient level anymore, only at the average overall study level. So these disadvantages are motivating us for going towards individual participant data meta-analysis. So in part 3 I'm going to talk about the rationale and advantages of this approach. So IPD means individual participant data. It used to be better known as individual patient data, but participant is a more inclusive term and should be therefore used. So this is a meta-analysis where rather than extracting aggregate data from publications, we're actually going to the original study investigators and asking for their raw individual level data. So this is at the participant level, so we'd have participant information about individuals' ages, their prognostic factors, their baseline characteristics, their outcomes at the individual level. So if you like, it's the original source material from which the aggregate data were derived for the publication. So an IPD meta-analysis is the synthesis in a statistical model of the IPD from multiple studies for the purpose of summarizing the evidence. This is increasingly relevant with the advent of stratified medicine, personalized medicine or precision medicine, where we're trying to go beyond the overall effect and tailor treatment decisions and healthcare for particular individuals conditional on their characteristics. And IPD meta-analysis projects have been advocated for quite a long time. They started to emerge in the late 1980s and the early 1990s. And this was accompanied by tutorial articles and articles suggesting why we needed IPD meta-analysis projects, why we needed data sharing, and why we needed to synthesize IPD rather than aggregate data. So papers by Leslie Stewart, Max Palmer, Mike Clark, all of these papers were very influential for setting the scene for people who were embarking on IPD meta-analysis projects in the 1990s. As I mentioned at the start, we have a book that's just been released by Wiley, and in there we have this this graph which we can see the number of publications of IPD meta-analysis articles over time. And with the advent of data sharing, in the last 10 years there's been an explosion of more IPD meta-analysis articles to about 300 per year around 2020 compared to 10, less than 50 before 2005. So with the advent of data sharing, and often funders stipulate that data should be shared after a study is finished, that IPD meta-analysis projects are going to become more and more common and are not going away. So here's an example of hypothetical IPD. Again, let's go back to our 10 hypertension trials looking at antihypertensive treatment. So when we have an IPD meta-analysis project and we've got our IPD and we've merged it together at the end into a single dataset ready for meta-analysis, we'll end up with a big dataset like this, where we'll have our trial ID, we'll have our participant ID, and then we'll have the information for each participant, such as which treatment group they were in, their age, prognostic factors such as their blood pressure before treatment, and follow-up information such as their blood pressure at 1 year. So rather than aggregate data where we had one row per study, now we've got one row for each participant in each study. So I've obviously had to omit data here, but like for study 1 had 1,454 participants, study 2 had 337 participants, so we have a row for each participant in each of these studies. So this is a big database. An area where IPD meta-analysis projects are particularly interesting is in prognosis studies. So here's an example of what IPD would look like in a cancer prognosis study, for example. We'd again have a study identification, we'd have a participant ID for each study, we'd have biomarker levels, we'd have other prognostic factors or adjustment factors such as age and stage of disease, so sort of routinely collected information. And then we'd have follow-up information. So in cancer prognosis studies, these would be time to event, so time to recurrence, time to death, disease status, and so forth. So we've got the richness of having the individual information. This leads to many potential advantages for those doing meta-analysis. So for example, we could use our own inclusion and exclusion criteria across studies, which may be different from the original trials or studies that they used. We could even reinstate individuals into the analysis who were originally excluded. We could observe and account for missing data at the individual level. So again, rather than being reliant on the analysis methods in the original studies, we can now apply maybe a more appropriate method for dealing with missing data at the individual level. We can verify the results presented in the original study publications, just to check that things were correct. We may even have longer follow-up information, so go beyond the study publications because we've got more data than was originally presented in the publication. When doing a meta-analysis, we want to make sure we have high quality evidence going into it, and so having the IPD allows us to inform risk of bias assessments, for example in regard to whether groups were balanced at baseline. And we can even, because we're doing our own analysis, we can improve upon the approaches used in the original studies and therefore maybe even reduce the risk of bias had we not had the IPD. Sometimes, for whatever reason, particularly in cohort studies, there may be some overlapping sets of participants across studies, and IPD would allow us to resolve that. We may have more outcomes available in our IPD than were available in the publications. And therefore we can overcome potentially the problems of outcome reporting bias by calculating results and aggregate data ourselves for those outcomes that we want. Sometimes, because of the way that IPD meta-analysis projects work and the collaborations and the dialogue involved with study investigators, we may get more IPD than we realise were available. So we may have IPD from unpublished studies, for example, and this may reduce the problem of publication bias. Standardizing this strategy of analysis across studies, so rather than maybe having some studies that use logistic regression and others use Cox regression, we can now standardize which analysis methods have been used, we can standardize how continuous variables were analyzed, and we can use more appropriate or advanced methods than were considered potentially in the original studies. So we could go beyond the assumptions made. A key one in time to event analysis is checking whether in a Cox regression model that the hazards are proportional, and maybe we could look at time dependent hazard ratios and maybe treatment effects and prognostic effects change over time. We could produce conditional treatment effect estimates or conditional results, that is results adjusted for prognostic factors. Again, this may increase power, reduce heterogeneity and be more appropriate for making decisions at the individual level. With stratified or personalised medicine in mind, IPD allows us to go beyond the overall treatment effect and look at subgroups of participants and see whether the treatment effect varies across particular individuals according to their characteristics. This is known as treatment covariate interactions. Away from randomised trials, we can even do better analyses in other areas, like looking at the accuracy of diagnostic tests, comparing tests, looking at the test results at multiple thresholds, not just those thresholds reported in publications. We could maybe validate prognostic models or prediction models, sometimes known as risk scores, and maybe even develop new models based on the information that we've got available. If we have multiple outcomes that are correlated at different time points, for example, then we could, with the IPD, we can account for the correlation at the individual level. This may lead to more appropriate analyses and more power, for example. So IPD meta-analysis projects are really aligned with the drive to reduce resource waste, to maximize the information coming from existing studies and existing data, and aligns with the drive towards personalized and precision medicine. But it's not perfect. It's going to be costly to do an IPD meta-analysis because you're going to need advanced expertise in database management, statistical analysis. You're going to need to engage many potential collaborators, and the time taken to discuss, clarify, check the IPD, and to disseminate and interpret the results with collaborators. It's time consuming, like I said, to check, harmonize and synthesize IPD. The IPD itself across studies is not necessarily going to be consistent, so people may have used different codings, they may have measured different things still, they may have used different measurement methods. So IPD doesn't solve those problems. It also doesn't solve a study being of poor quality. You can't resolve the way that people did the randomization, for example. Whereas standard meta-analysis methods are relatively straightforward, an IPD meta-analysis would need more sophisticated methods and therefore more specialist expertise in statistics, which may not be available. Dealing with complexities such as missing data are also important to consider. So in part 4, I'm going to talk about notable examples of success stories in IPD meta-analysis projects. So here's a well-known example, which was an IPD meta-analysis conducted by the Early Breast Cancer Trialists Collaborative Group. They obtained IPD from 55 randomized trials, including 37,000 women with early stage breast cancer. Incredible achievement to get IPD from those studies. And they used that IPD to examine whether the benefit of tamoxifen therapy varied according to estrogen receptor status, ER status. And their IPD meta-analysis did demonstrate strong evidence, backed up by biological rationale, of a larger treatment effect for the ER positive group. So here we have the meta-analysis result for the ER positive group. Clearly the hazard ratio is much less than 1, whereas for the ER negative group the hazard ratio is very close to 1 and there's a lot more uncertainty. So this is one of the major reasons why tamoxifen treatment is generally only given to those that are ER positive. In that previous example, there was a perhaps artificial dichotomization into whether estrogen receptor is positive or negative. But with the IPD, you can actually go beyond simplistic assessments of continuous variables like that and use a more appropriate analysis considering those variables as continuous. So for example, Wang et al. And then myself and colleagues a bit later used IPD from 10 trials of antihypertensive treatment to assess whether the treatment effect differed according to age. And there is a sort of a U-shaped relationship where we see that relative to individuals aged 55, the treatment effect for those that are younger gets more positive here. So this is the difference in the treatment effect. So a positive means the treatment effect is doing less well. Remember that on average the treatment effect was about -10. So here we have about +5 compared to that. And this confidence interval is mostly all above 0. So strong evidence that those younger participants, their treatment effect is not as good, but still useful, compared to those that are age 55. Whereas those that are age 55 and older seem to have reasonably similar treatment effects, and there's no evidence of a difference compared to the 55 group. what we're doing here is also looking at a non-linear relationship, and you just couldn't do this without IPD. You couldn't do this because the aggregate data wouldn't be reported to allow you to look at non-linear relationships like this. Moving away from randomized trials, let's look at test accuracy at multiple thresholds. So this is where we're looking at the sensitivity and the specificity of a particular test. And when we do that for a continuous test, we need to choose a cutoff, a threshold value. So on this graph, which is known as an ROC curve, I've got threshold values from 7 all the way down to 15. So 7, 8, 9, 10, and 12, 13, 14, 15 for the PHQ-9 score, which is looking at depression and diagnosing depression. Now, without individual participant data, you're reliant on the original studies, what they reported and which thresholds they used, and this leads to different thresholds being reported in different numbers of studies. And so what we should be seeing here is a monotonic ROC curve like what we get with the IPD, but without the IPD, the results from the individual studies, when they're pulled together, we get this jaggedy and non-monotonic curve, which is inappropriate, and it's because different thresholds have different numbers of studies contributing to them. But with the IPD, we have the original continuous test value, and therefore in every single study we have the ability to work out the threshold for every single threshold of interest, the sensitivity and specificity at that point. So we have this nice appropriate curve. What about added prognostic value? Well, here is an example looking at the IPD meta-analysis of 17 published and unpublished studies, 3,200 participants with non-small cell lung carcinoma, and the question was, is microvessel density a prognostic factor for death? IPD enabled results to be derived for each measurement method. Here, this is the all vessels method. We could adjust for well-known existing prognostic factors of age and stage of disease and analyze microvessel density as a continuous variable, here assuming a linear relationship. So when we did this in an IPD meta-analysis, sorry, I want to say we, I mean Marilena Trevelli, Doug Altman and colleagues, they put the results together, and we see that the hazard ratio for a 1 unit increase in MVD is 1.03 and the confidence interval overlaps 1. So there's no clear evidence that microvessel density is adding prognostic value over and above the known prognostic factors of age and stage of disease. Interestingly, this completely contradicts an earlier meta-analysis based on published aggregate data that concluded microvessel density was strongly a prognostic factor. But that previous published aggregate data meta-analysis didn't adjust for age and stage of disease, was reliant on the analysis method in the original publications, which all use different cut point levels, and therefore couldn't handle microvessel density as a continuous variable. So the IPD meta-analysis is much more reliable evidence here, contradicting that of a previous aggregate data meta-analysis. And the last example I want to showcase is the ability to validate and compare prediction model performance. So prediction models are where you predict the risk of outcomes for individuals based on their characteristics. And here I'm looking at an IPD meta-analysis to validate the performance of Model 1 and the performance of Model 2 in terms of a measure called the calibration slope. So calibration of observed and predicted risks, we would like that to be 1 to give it some indication that the model is well calibrated in terms of predicted risks in new individuals. And this was examined using IPD meta-analysis with IPD from 8 countries. We see that Model 1, when we do our meta-analysis of the calibration slope across studies, we get that the summary calibration slope is 0.99. So on average, very close to 1, which might suggest that this model is doing very well. But you can clearly see there is a lot of heterogeneity. For example, in study 3, there is substantial miscalibration. Study 8, substantial miscalibration. So this average result is an incomplete picture. Model 2 also has a summary result close to 1, a narrow confidence interval, but there's very little heterogeneity. In every single study, the calibration slope is very, very close to 1. And therefore, on average, Model 2 is as good as Model 1, but also it's more consistent in performance across settings. Now, without IPD, you wouldn't be able to assess calibration at all, because you need to assess calibration at the individual level, and you wouldn't have that at the aggregate level. Okay, so there's some really nice examples of why IPD meta-analysis projects are important. And I'm just going to finish with some key steps involved in IPD meta-analysis projects in case you're thinking of undertaking one in the future. The first question you need to ask yourself is, do I need IPD for meta-analysis? So you need to ask questions such as, well, what is your research question? Has a previous review been done before to answer this question? What aggregate data would be required to answer your question? Is such aggregate data available in the majority of studies? If it isn't, then if you did get the IPD, would it allow you to calculate that aggregate data directly? Or is the IPD would it not still be sufficient? How much IPD could you realistically obtain? Do you have any agreements with people already? Is it plausible to get it? If you got it from the majority of studies you wanted it from, would that still be giving you enough power to answer your research question? How long do you think it's going to take you to get the IPD? Indeed, do you have the resources for obtaining it, for collating it, for checking it and managing it? And do you have statistical expertise and software to analyze the IPD? So these are big questions in terms of whether the approach is achievable and necessary. And it's aided by collaborating groups getting together, different disciplines working together, leaders in the field being involved and therefore giving their support and weight to the project, and of course research funding. The more nuanced your research question, the more likely you are to need IPD. So if you're just interested in the overall average treatment effect, it's likely that those traditional reviews such as those done by Cochrane would be sufficient based on aggregate data, because most studies focus on publishing information about the overall treatment effect. However, if you want to go beyond that, to maybe adjust for prognostic factors, to look at the effects in subgroups, to maybe compare subgroups, look at interactions. If you're looking at doing more complex analyses, such as analysing nonlinear relationships, maybe investigating non-proportional hazards, maybe multiple time points, or if you're going even well beyond the original study in terms of wanting to use the IPD for a different purpose, like validating a prediction model, then as you come down here, this is more likely to need the individual level data rather than anything from the publication. Also, IPD may allow you to improve quality. So even if you're interested in the overall treatment effect in a very controversial area, for example, it might be that people want to do an IPD meta-analysis to check the quality, to improve your risk of bias assessments of studies, to change the inclusion criteria, to check assumptions, to include more outcomes. So even for more simple questions, IPD can make things better and improve quality and transparency. Maybe the analysis methods within parameters were not appropriate and having the IPD would address that. Maybe the data is fine, but you're trying to estimate something like a conditional treatment effect, whereas the original study focused on the average overall treatment effect, a marginal treatment effect. The current reporting standards within primary studies may be a problem to getting aggregate data, and therefore IPD would address issues of reporting biases. If you've decided that an IPD meta-analysis project is important, then you need to think about, okay, I want to get the IPD, but will the IPD itself be of sufficient quality? Will it provide the required participant level characteristics that I'm interested in? So if you're interested in some new fancy biomarker, you need to check that that biomarker has been recorded by those studies. Does the IPD record the outcomes of interest to you? Do you have, with the potential IPD you can get, reliable statistical power to address your research questions? Therefore, careful planning and preparatory work is needed to ensure the IPD meta-analysis project is achievable. And this is an important point to take home. IPD meta-analysis projects take a long time, often up to 2 years to complete. They require specific research funding. It's not something to be done in your own spare time, which perhaps sometimes traditional reviews have been done. And it's going to need a big team with broad skills, much broader than the conventional systematic review, because you're going to need, for example, more statistical expertise and more experience in database and management of participant level data. So if you're setting up your IPD meta-analysis project, develop your project scope, establish a central research team and advisory group, seek in principle agreement to collaborate from study investigators. So don't embark on it blindly. Get in touch with people beforehand to make sure that it's reasonable that you will be able to get the IPD. Identify where you're getting your IPD from, and maybe there's other sources, maybe other databases you can get IPD from. You're going to need to develop data sharing agreements, apply for research funding to support your project, and make sure that ethical approval is in place or to have an exemption if that's appropriate. By a central research team, we mean members with experience of successfully completing an IPD meta-analysis project, such as those with advanced statistical knowledge, experience in managing and coding IPD and getting IPD, and the team needs to have strong communication skills because there's going to be a lot of back and forth between the original study investigators and the IPD team in terms of getting the IPD across, resolving queries and disseminating findings. It's really helpful to establish a PCOS framework for your study. So this is particularly for trials, but there's been extensions for other types of studies. So what's your population of interest? What's your intervention of interest, the comparator? What are the outcomes of interest, and what's the design of interest? Collaboration is a key word. Most IPD meta-analysis projects that do well are collaborative in nature. They involve partnerships and dialogue and open transparency with the original study investigators. At the moment, there's a drive towards putting IPD in repositories or data sharing platforms. And this is exciting. It's an alternative and maybe will complement collaboration with trial investigators. But you need to be cautious because the IPD in a data repository may not be the complete IPD from the original trial. And you don't want to stop collaboration and communication with the original trial investigators. So it's still helpful to communicate with the original trial investigators, even if the IPD ultimately does come from a data sharing platform. Advisory groups are important to include wider clinical knowledge and methodology expertise and to get patient and public involvement. And patient and public involvement and engagement is helpful throughout the whole project, from developing the research questions to, of course, interpreting and disseminating your results. It's also helpful to set up a protocol, so register a protocol and publish your protocol telling the reader about the objectives, the inclusion and exclusion criteria, the outcomes to be evaluated, what participant level covariates are of interest, what the process is for data transfer, collection, coding and checking, how you will examine the quality of existing studies and their IPD, what statistical methods you will use, and maybe have an analysis plan that's separate to the protocol itself. So the overall aim should be to maximize the quantity of high quality IPD in order to limit bias and reduce uncertainty of results, and to make sure that your planned IPD meta-analysis project can be reliably completed in the available timeframe. A huge and often underappreciated aspect of IPD meta-analysis projects is the time needed to get the IPD and check it and clean it and harmonize it. So IPD needs to be transferred and stored securely. It needs to be used carefully and appropriately in order to respect patient confidentiality agreements, and it needs to adhere to the contractual agreements that were put in place with those that provide their IPD for your project. Variables that are supplied in their IPD will often need to be recoded, redefined, and put into a common format to enable the subsequent meta-analysis of the IPD. This will take considerable time, and often there are queries and issues to be resolved with the original trial investigators, and sometimes errors are found, and therefore there's a loss of back and forth. It's helpful to have a detailed log of all your changes and transformations made to the IPD that was provided, so that people can independently check what you've done, and it's a transparent and reproducible process. This is an interesting letter that was published in the BMJ, and I really include it here just to emphasize to people that collecting IPD is not a quick job. So this is just for one study, and it says, why did it take 19 months to retrieve clinical trial data from a non-profit organization? And in the letter it says, the emails we received during the prolonged exchange were all friendly, and the individuals involved were helpful and understood the need for data sharing, but they were hampered by inflexible, formalistic and slow bureaucratic procedures. Since our first inquiry, we communicated with 4 people, sent 25 emails, filled in 4 data use agreement forms, and waited for 1 year and 7 months. That's just for one IPD. Now, that is an extreme example potentially, and there are obviously examples where it's much quicker than that, but it just goes to show that even in the era of data sharing, that obtaining IPD may not be straightforward, and you need to bear that in mind when setting up and embarking on IPD meta-analysis projects. A key step once you've got the IPD is to examine risk of bias. So risk of bias of a trial is well known in traditional reviews, but here we're talking about risk of bias not just for the study, the trial, but also for the IPD itself. So this initially is going to be based on the information from publications, but then you may get additional information provided by study investigators, and then once you've got the IPD, you may be able to refine or update your risk of bias concerns based on what you see in the IPD. So for example, by re-including some participants in the IPD, you may be able to reduce concerns of risk of bias. Conversely, you may identify some strange randomization approaches have been conducted, maybe in extreme situations where all the treatment group participants are randomized first, the first 6 months of a year, and then the second 6 months of a year, the control group participants are randomized. There may be extreme baseline imbalance, which is worrying, and therefore IPD may reveal some risk of bias concerns that you hadn't identified before having the IPD. So IPD can help with risk of bias assessments. Once the data are checked, harmonized, risk of bias is concluded, we need to do meta-analysis. So meta-analysis methods for IPD usually use either a 2-stage or a 1-stage approach. And they're followed by sensitivity analyses. Like any good statistical analysis, there are assumptions, and we need to examine the sensitivity of results to those assumptions. So briefly, a 2-stage approach involves, in the first stage, analysing each study separately using an appropriate analysis method according to that study and its design. For example, in a randomised trial, we may apply a regression analysis in each study to produce a treatment effect estimate and its variance. For example, using a linear, logistic or Cox regression, maybe adjusting for prognostic factors. So in the second stage, we then take the aggregate data that we've produced, such as the treatment effect estimate and its variances, and pull it together using the traditional meta-analysis methods like an inverse variance weighted meta-analysis, producing our summary results and forest plots, for example using a common effect or a random effects meta-analysis model. In a 1-stage approach, the IPD from all studies is analysed all together in a single analysis, accounting for the clustering of participants within studies. So what we're talking about here is an extension of standard single study analyses to allow for clustering and heterogeneity. So we're talking about generalized linear mixed models, survival models with random effects terms such as frailty models. And so we're simultaneously analysing all the IPD in one go. 1-stage models utilise a more exact statistical likelihood because you avoid the 2 steps, you analyse it all in one go. Therefore, you don't need assumptions about whether effect estimates are approximately normally distributed with known variances. And so this is advantageous when most studies have few participants or events. However, in other situations where most studies are reasonable in terms of number of participants and events, say more than 30, then 1-stage or 2-stage approaches should give similar results, especially if you're using the same assumptions and estimation methods. So for example, if you're assuming a common effect model or a random effects model, you need to assume it's the same in both. If maximum likelihood estimation is used in both, then you're going to get similar answers to the summary results, as long as you don't have sparse data in the individual studies. Generally, I recommend using a 2-stage approach because this is very familiar. It will give you some clear visual summaries such as forest plots. It allows you to include in the second stage aggregate data from other studies that didn't provide you their IPD, and it's generally quite quick. For example, there's a brilliant package called IPD Metan in Stata, which was written by David Fisher from UCL in London, and it's a brilliant package that does the 2 steps for you and produces lovely summary results and forest plots. Lastly, once your results are obtained, the final step is to report and disseminate your findings. And this should be planned from the outset. It's not just a journal article that's important. It's reporting and dissemination to a whole range of potential stakeholders, including patients and policymakers. The main things to note is that for randomized trials, PRISMA IPD has been published, and this is a reporting guideline providing a framework to help authors describe elements of IPD meta-analysis projects in their journal articles. But like I said, it's not just about journal articles. There's a lot more to disseminating results that should be considered to make sure the IPD meta-analysis projects have the greatest impact on those that the results could be influencing. Lastly, I want to finish by just telling you about our new website and our new textbook. So firstly, our new website, ipdma.co.uk. This is a central resource for healthcare researchers to go to if they want to find out about IPD meta-analysis projects. So if they're new to the field or they want to identify recent developments, then this is the place to go to. We're promoting good practice, giving entry-level information, important steps and principles that relate to the chapters in our book, giving reference lists, videos of new seminars, the statistical software code and information about software packages, information on training courses. So for example, at Keele University we run a number of training courses, one on statistical methods for IPD meta-analysis and one on the more general overall methodology and principles for doing IPD meta-analysis projects. So you can find information about those courses there. So please check ipdma.co.uk out. And then lastly, our book, Individual Participant Data Meta-Analysis, a Handbook for Healthcare Research, came out in 2021, published by Wiley, and this is a comprehensive introduction to the whole process of doing an IPD meta-analysis project. 18 chapters, over 500 pages. We've collaborated with researchers who've got substantial experience in doing IPD meta-analysis projects, and we cover the basics, the how to get up and running, the planning, the undertaking, the statistics, and then novel topics, not just randomized trials, but also diagnostic studies, prognostic studies and prediction studies, going beyond overall treatment effects, looking at effect modifiers, test accuracy, prognostic factors, risk prediction models. We hope you'll find it really, really useful, and we very much welcome your feedback in due course. So thanks very much for listening to this talk. I hope this has been a useful introduction to IPD meta-analysis projects. Any questions or queries, please send me an email or DM me on Twitter. Thanks very much.